Hello guys, welcome to a new video for the Brain Bee. Today we're going to be talking about basic neurobiology, which is very important because it really tells you how the neurons work, right? And understanding our neurons and those very specialized cells are going to be very beneficial to understanding the bigger things and concepts in neuroscience as a whole, right? So let's get started. As I said already, the neuron is the basic unit of our central nervous system and peripheral nervous system, which means the neurons are the main cells that we care about for the most part when we talk about the central nervous system and the peripheral nervous system. That is not to say that we don't have other important neurons, but for the most part, all the other neurons are helping neurons in their function, right? So there are different parts of a neuron. As you can see here, there's the axon hillock, the axon, the myelin, the node of Ranvier, the dendrites, and the axon terminal. But let me show you a schematic of what a neuron looks like, and then we'll go a little bit over what each part does. So here you have a neuron, right? And all the parts are being labeled. And so each particular part is very important. So let's talk about the dendrites. These dendrites are what allow other neurons to communicate with this neuron that we have here. So that is the part or the postsynaptic component that receives information from the other neuron and receives information such as neurotransmitters and chemicals, right? You have the axon hillock, which is where the action potential generates, and we'll talk about the action potential here in a second. You have the axon, which is not labeled here because it's covered by myelin. So these yellow blobs or yellow portions are myelin sheath, and the axon would be underneath that, right? And so the spaces in between each portion of myelin is what you'd call the nodes of Ranvier. And the nodes of Ranvier are very important because that allows for saltatory conduction, right? And in the action potential, we have what we call the refractory period. So I want you guys to remember this refractory period because it'll come back in the action potential portion. But the longer the refractory period, the longer the space in between the segments of myelin. And it's particularly important because these segments of myelin, or the nodes of Ranvier, are very enlarged in demyelinating diseases, right? So then the conduction of the action potential won't be as effective. And so if the axon hillock is where the action potential generates, then the axon is where the action potential travels. And that traveling of the action potential is what ends up reaching the axon terminals. And the axon terminals release chemicals that we call neurotransmitters. And these neurotransmitters allow the communication from a neuron to another neuron, or a neuron and a tissue, or a neuron and gland, right? So this is very important, right? So I want you guys to take maybe 2 minutes to quiz yourselves. What is the axon hillock for? What is the myelin sheath for? What are the nodes of Ranvier? What are the dendrites? And what are the axon terminals? Good. So now that you know what the axon terminals are actually releasing, neurotransmitters, let's look at some of those neurotransmitters. Neurotransmitters come in 2 ways: excitatory or inhibitory neurotransmitters. The excitatory neurotransmitters elicit or enhance an action, right? They stimulate something. They make something happen. And the inhibitory obviously do the exact opposite thing, right? So they reduce an action, they diminish it, or they prevent it from happening. Now, these are the most common neurotransmitters that you'll find. Glutamate, dopamine, acetylcholine, histamine, and epinephrine are on the excitatory end. And GABA, glycine, serotonin, and norepinephrine are on the inhibitory end. Now, let me give you a few examples of what these neurotransmitters are for. Glutamate. Glutamate is the main excitatory neurotransmitter in the body. And what that means is that it is the most commonly used to excite other neurons and generate other action potentials, right? It's also found in the spinal cord very heavily for the interneurons to work with each other. And it's very important for the function of the entire nervous system. So a downside of glutamate is the excitotoxicity, right? And that is because Too much glutamate can actually cause neurons to fire erratically and lead to such things like epilepsy or even migraines. Dopamine is very, very important because it helps with some movement. As you may recall, if you have Parkinson's disease, you have a loss of dopaminergic neurons in the brain, especially in the midbrain, and the substantia nigra of the midbrain, right? And dopamine is also important for the reward pathway. So if you guys remember your reward pathway, it's highly active due to dopaminergic neurons, and that is because instant gratification or things that you enjoy doing, such as eating, right, release high amounts of dopamine, which are very, very important for that feel-good moment. Acetylcholine is very helpful in memory and skeletal muscle contraction, right? So the acetylcholine helps muscle contraction in the skeletal musculature, but it actually promotes relaxation in cardiac muscle. So you can see here how one neurotransmitter has different functions depending on the location. So again, acetylcholine is very, very good for muscular contraction in skeletal muscle, but for muscle relaxation in cardiac muscle. Histamine is very important because it plays a huge role in your waking and arousal. So after a long sleep, some of the components that are released to keep you awake and make you awake are histamine, and epinephrine is important because it controls your heart rate, right? So it's very, very important. And then on the other hand, your inhibitory neurotransmitters are counteracting all of your excitatory neurotransmitters to keep a balance. And that balance is very, very important because it's what helps us really function properly. So now you know a little bit about neurotransmitters. Other released chemicals, well, peptides, something that a lot of people don't know, or somebody that at high school levels don't know, so which is why you guys are far more advanced, is the existence of peptides. And for example, substance P. Substance P increases your sensitivity to pain. So the more substance P that you have, the more pain that you release, or that you feel, sorry. And enkephalins and endorphins are your endogenous or naturally synthesized painkillers. So the body has a system to reduce pain, right? Imagine if you were walking around and you stub your toe in the table and you didn't release these enkephalins or endorphins, you'd feel that pain for a very, very long time, right? So such chemicals help you with reducing the pain sensation. So these are very, very important. And then furthermore, the neurotransmitters can be divided into different kinds. So biogenic amines and amino acids. Your biogenic amines are pretty much everything that ends in NE. So acetylcholine, dopamine, norepinephrine, epinephrine, serotonin, right? With the exception of glycine, because glycine falls under what we call amino acids. And some of the amino acid neurotransmitters we have are GABA, glutamate, and obviously glycine. So now we know that the chemical components are neurotransmitters, and we know that these neurotransmitter release is what actually allows the communication from cell to cell, right? But what releases these neurotransmitters is a bigger question, right? They don't just release themselves. There has to be some sort of stimulus, and that stimulus has to trigger a response that ultimately leads to the release of neurotransmitters. So let's look at the action potential. This curve is very, very important. Believe it or not, guys, this curve is taught at a college level in some of the physiology classes, neurobiology classes. So if you guys are able to understand this action potential curve, you guys are going to be fairly advanced in your education. So let's take a peek at it. I want you guys to look at some values that are very, very important, right? So I want you to look at negative 90 millivolts, negative 70 millivolts, 55 millivolts, and plus 40 millivolts, right? These are going to be very, very important. So let's see what happens during an action potential. You have some stimulus, right? And the stimulus, it's the thing that is going to actually make the neuron fire. And by fire, I mean release the action potential. So for that to occur, you have to beat the threshold. So negative 70 is what you call your resting membrane potential, right? That is without any stimulus. That is where the cells or the neurons are naturally at. And then you have a stimulus that has to be good enough to beat or reach this minimum threshold of negative 55 millivolts to actually go all the way to plus 40 millivolts to elicit the action potential, right? If you do not reach the minimum threshold of negative 55, you're gonna have what you call failed initiations. And that is because neurons work on a yes or no response. So either they fire or they don't. There's no in between. There's always all or none response, right? And so this depolarization going from negative 70 to plus 40 is what actually allows the action potential to occur. This is the action potential. But the cell doesn't stay there forever, right? It has to go back, or it has to repolarize so that it can be elicited again and then fire again, and so on and so forth repeatedly. However, upon repolarizing, it could reach negative 90, which is a little bit lower than your negative 70, which is what we call the resting state. So if it reaches negative 90, is what you call hyperpolarized. And this hyperpolarization comes with a refractory period. And during that refractory period, the neuron will not fire under any circumstance. It lasts a few milliseconds, but it's very, very important that you guys understand that if the cell has been hyperpolarized, it will not fire anymore, right? So now you guys know this action potential curve and how it works. However, there are some very important chemical components that go along with this action potential. So again, I want you guys to focus on your negative 90, your negative 70, and your negative 55, right? And your plus 40. Let's look at some of the ions that are involved with this. First and foremost, the neuron is able to maintain a resting potential because of the sodium-potassium pump. So this sodium-potassium pump allows the exchange of sodium and potassium ions, right, to maintain it at a resting state. Once it gets stimulated, it goes to a positive because there's an influx of sodium ions into the cell. Then it reaches the action potential, and to repolarize or go back down, it has to have potassium ions exit the cell, right? Now, if you look closely, sodium and potassium both have a positive charge. So how come is it that you can go from a positive plus 40 when the action potential happens to a negative 70 to repolarization, right? And that is because potassium is a much more positive ion than sodium. And when potassium leaves the cell, it makes it so that now you have a negative charge inside the cell, right? So it is because of your periodic table trend and the positivity of this potassium ion that when it leaves, it's able to go back to your resting membrane potential, right? And so I have here an axon. So I've drawn an axon here, right? So usually the intracellular space of the axon, it's negative. So the charge inside the cell is negative during the resting membrane potential, which makes sense because at the resting membrane potential, we said we have a negative 70 millivolts, right? So it's logical that the cell inside will be negatively charged. Then it depolarizes, it goes through the action potential, it depolarizes, and it changes charges. Now the inside of the cell is positive, and the extracellular fluid, it's negative, right? And for the cell to go back and repolarize, so to go back down to where we were before, that means that the potassium has to exit the cell, right? Bringing it back to our negative concentration, right? And then if it hyperpolarizes going to negative 90, the sodium-potassium pump acts again to bring it back to that negative 70 so that it can be stimulated and the action potential can be elicited once more, right? So now you know the chemical aspects of the action potential, which are very, very important. So here again, we have a nice little GIF or a GIF. I don't know what you kiddos call it nowadays. But it's essentially explaining how that change of charge between the intracellular and the extracellular components is occurring for the action potential to occur, right? So again, if you remember your neuron anatomy, this is the axon hillock. This is where the action potential generates. This is the axon right here. This is where the action potential is going to travel. And in this particular one, we're not myelinated. But if you were myelinated, then you would know it had some oligodendrocytes or some Schwann cells, around it, right? And that is the action potential on the axon itself. So we've gone over the electrical components of the neuron and how that chemical and ions interactions allow for the release of neurotransmitters. So let's see how the neurotransmitters end up reaching the adjacent neuron, right? So let's look at the chemical communication. After the action potential happens, the vesicles inside the terminal boutons, or the axon terminal, right? These vesicles contain the neurotransmitters, which are going to be transported into the synaptic cleft so they can bind into the postsynaptic receptor, right? So essentially what's happening is that the action potential, it's allowing the vesicular transfer to occur to release the neurotransmitters so that they can elicit an action on the adjacent cell, right? And so if you look here, here's your synaptic vesicle, right? And it's carrying all these little neurotransmitters, the green buttons. The neurotransmitters then reach this part, right? And they get released so they can act on the receptor on the postsynaptic space, right? Now, when you think about how the receptors and the neurotransmitters work, right? So the receptors are very, very selective. So if this receptor right here, it's for acetylcholine, for example, then acetylcholine has to have a structure that will bind specifically to this receptor, right? So if you had a receptor here, for example, for glutamate, you can see that the difference in the receptor, right? So acetylcholine wouldn't be able to bind because glutamate has a specific affinity for that receptor and vice versa that is very one-to-one, right? So it's very, very important that the receptors are present for the receival of the neurotransmitters, right? So just as an example of how this could be affected. In certain diseases, like myasthenia gravis, which is an autoimmune disorder, your immune system attacks these receptors, and when you lose those receptors, then acetylcholine doesn't bind properly in myasthenia gravis, and then you don't have muscle contractions, right? So that happens because now you don't have enough receptors allowing for the acetylcholine be released to bind. And if acetylcholine doesn't bind, it doesn't elicit a reaction, and then there's no contraction of the muscle, right? So something that we can do is to give medications such as acetylcholine esterase inhibitors. And acetylcholine esterase breaks down acetylcholine. That's why it's called acetylcholine esterase. So every time you hear esterase, it means that it's breaking down something, right? And we're talking about neurotransmitters. So in myasthenia gravis, as I said before, what you want to do is that if there's not enough receptors for acetylcholine to bind, you want to make sure that acetylcholine stays in the synaptic cleft long enough without breaking down so that they have more time to reach the receptors. But you're not able to do that because we have acetylcholine esterase, which breaks down the remaining acetylcholine in the synaptic cleft. So the acetylcholine esterase inhibitor will inhibit that breakdown of acetylcholine and will give more time for the acetylcholine to find the receptors and bind to it, right? So this is essentially how the chemical communication works, right? So now you know how the action potential works. That is the electrical component, right? And now you know how the chemical aspect works. And putting those two together, now you have the full view of how the neurons work, right? So it's very, very important that we understand this neurobiology going on. So now let's talk about different types of cells. Neurons, being the most important cell in the central nervous system and the peripheral nervous system, need some help. And we have what we call glia. And glial cells are really the most important aspect that can potentially aid a neuron. Because in the central nervous system, we have astrocytes, oligodendrocytes, microglia, and ependymal cells. But in the peripheral nervous system, we have satellite cells, Schwann cells, and microglia. And although the functions may be very, very similar, it's important that we distinguish between the both, because the central nervous system, it's, as you guys know now, the brain and the spinal cord. And the peripheral nervous system is like the distal nerve, so like the distal portion of your body, which would be your limbs, your legs and arms, right? And also a little bit more advanced, which we'll get to that later, would be the cranial nerves. The cranial nerves are considered part of the peripheral nervous system, right? So now let's take a peek at what they do, right? So the ependymal cells, which look something like this, line the cavities of the ventricles. And the ventricles are a cavity in the brain that hold CSF, cerebrospinal fluid, right? They're also in the central canal. And the ependymal cells help you with the direction of CSF. So I want you guys to keep in mind that CSF is not stationary, it moves, it has a flow, right? And part of why it moves is because the ependymal cells have what you call microvilli, and these microvilli allow for the direction of CSF within your ventricular and spinal cord, right? The oligodendrocytes, which are still a central nervous system, right? They myelinate the axons. The astrocytes, they provide nutrients to the cell. So they bring in glucose and other very, very important molecules for the cell. So they pretty much help you nourishing the neurons. The microglia are a type of macrophage that helps cleaning up debris. So whenever you think about debris, think about what happens if you cut a neuron, right? Obviously there's going to be some debris. So think about it as a glass bottle. If the glass bottle was your neuron and you break it, there's a lot of tiny pieces of glass that are going to go everywhere, right? And so the microglia would come in and pick those up to prevent that debris from hurting other neurons or releasing toxic contents into the brain, right? Then your satellite cells and your Schwann cells are in the peripheral nervous system. So for example, your Schwann cells do the same thing that an oligodendrocyte does, but they only myelinate one neuron at a time. So as you can see here in this picture, the oligodendrocyte has a lot of projections. In this particular view, it's only putting all of its projections into one axon. But if you were to imagine another axon here, the astrocyte would be able to then myelinate this axon right here with its projections. However, the Schwann cell, it's a 1-to-1 ratio. So it only myelinates one axon at a time, right? So now we know, right, what some of the glial cells do. So here's another little chart that will help you learn this a little bit better, right? So just make sure you know the difference between the glial cells in the central nervous system and the glial cells in the peripheral nervous system. Very, very different components, yet very, very similar in function. So it's important that you're able to determine which one belongs where. And lastly, we're going to talk about the types of neurons. So the types of neurons, right, are very important to know because depending on the type of neuron, you can also find different functions for them. So we have unipolar neurons, bipolar neurons, pseudo-unipolar neurons, and multipolar neurons, right? Unipolar neurons are not as common, but they're very important for sensory perception. Bipolar neurons are very, very important, and they're mainly in your retina. So if you guys remember when we talked about senses, we talked about the different types of layers in the retina, and bipolar layers is one of them. So they're very, very important for visual processes. Pseudo-unipolar, they're mostly the dorsal root ganglion, and they're sensory, so they carry sensory information from the periphery into your spinal cord, and that can be processed. Multipolar neurons can mostly be seen as interneurons, or they innervate muscles, and they're usually your typical neuron. So when most people think about a neuron, they think about drawing it in the way that a multipolar neuron looks, right? And again, these are very important to know because depending on the neuron, it can give you a hint or where in our central or peripheral nervous system we are, right? So it's very, very important. So that is it for today. For questions and answers, so I would practice and ask among yourselves, what are the components of a neuron and what do they do? So what are the axon hillock, the axon, the Schwann cells, the dendrites, and the terminal buttons? How does an action potential work? Some of the crucial voltages of an action potential, the ions involved in the action potential, what ion is important for depolarization, repolarization, and where is the sodium-potassium pump active, right? Lastly, what are the types of glial cells that we have and where are they found? Are they mainly in the central nervous system, mainly in the peripheral nervous system? So if I tell you that there's a Schwann cell myelinating an axon, is that on the central nervous system or the peripheral nervous system, right? And finally, where are the types of neurons that we see? If I say that we have a pseudo-unipolar neuron, where is that located? So if you guys can answer these questions, you'll be very proficient with some Neurobiology 101. And we hope to see you soon for the Brain Bee.